We get started. Good afternoon, everyone. Welcome to another Terasaki seminar series. Today we have Professor Fariba Dehghani. She is currently the Director of Bioengineering, Director of Center for Advanced Food Engineering, and a former Director of an Australian Research Council for Food Processing Training Center at the University of Sydney, Australia. She has won many awards for her research. She was recognized by Engineers Australia as one of Australia's top engineers for innovation. She also received 2017 award for excellence from Australia and New Zealand Federation of Chemical Engineering, and also a UNSW Alumni Award of Excellence in Engineering. The University of Sydney Vice Chancellor Awards for her outstanding supervision and excellence in research, engagement, and innovation. She has completed her PhD degree in UNSW. After that, she received the prestigious Australian Research Council postdoctoral fellowship and continued working at UNSW as a senior research fellow and a senior lecturer. After that, she joined the University of Sydney in 2006, where she established a state-of-the-art bioengineering laboratory, enabling multidisciplinary research collaboration. She promotes entrepreneurial engagement, resulting in 4 startup companies being established by her research members. The floor is yours, Fariba.
Okay. Thank you, Mehmet, for your introduction, and also thank you for inviting me. This is a privilege to join your program today. Our Bioengineering Center is located at Sydney University, which is very close to the Sydney downtown in here. And I hope in near future you can join us and enjoy this beautiful city and many sightseeing in here. Our vision for research is to promote human well-being by designing materials for the prevention and treatment of diseases, and also developing sustainable nutritional food that can promote health while minimizing food waste. In terms of material, we are designing different types of material that can be used as an implant, or it can be used for tissue engineering or drug delivery, also for designing biosensor for medical application or other diagnostic purposes. Also, we work a little bit about in the area of organ and chips, and other area of our research in bioengineering center is focuses on designing nutraceuticals and sustainable food products. And I would like to give you examples of different parts of our research, mostly related to designing material, and I will touch base a bit about our research in the food area as well. We are all familiar with the polyester, which are broadly used as a from different parts of medical application, for example, as a sutures, or even for bone implant application. We are working with another class of the polyesters and designing next generation of polymer ceramic material for bone implants. This polymer is called polypropylene carbonate, which is made from carbon dioxide and propylene oxide. And the good thing about this material is very easily degrade to CO2 and water, and it is very good for both packaging application and also medical application. We can make film, fibers, and different structure on the top of this material. And it will be easily mixed with starch and natural polymers. And the degradation of this polymer is very similar to the polylactic acid, as you can see in here. And if you want to increase the degradation rate, we always can increase the amount of starch in this polymer, and the degradation rate can increase. Another feature of this material is, while it has very good mechanical property compared with PLA, when you add it to the biological system, it doesn't actually change the pH compared with PLA, which usually cause, pH drop by degradation, and it can cause necrosis and negative impact on surrounding tissues. This has been proven by doing some animal study. In here, you can put, for example, PPC-starch in this as an implant, and we can see that there is no fibrous formation or any immunological response toward this material. But when we compare it with PLA, after 8 weeks, we can see some fibrous tissue formation around the PLA because of these properties of polylactic acid. And we compare the property of this material with commercial product which is in the market, PLA calcium phosphate screws that usually they use for bone implant application. And we could see that both the PPC-starch bioactive glass and PLA calcium phosphate can degrade. And the degradation of the PPC-starch sample was much higher than PLA, almost double. And we could see that while PLA doesn't integrate with the surrounding tissue in 3 weeks and 12 weeks, we can see that this material can start to integrate with the surrounding tissue. And this histology result in here shows that PLA doesn't integrate with the tissues around. But for PPC, we can see that it's resulted in healthy cellular response after 3 weeks. And within 12 weeks, we could see that surrounding bone started to invade into this implant, and it has very good biocompatibility and can result on bone regeneration. The outcome of this research led to establishment of a startup company, SDIP Innovation, by Iman Manavi and his partner, and they received significant amount of funding from New South Wales government to bring this new product to the market for bone regeneration application. And now this product is in the clinical study, hopefully come to the market soon. The next material that I would like to share with you is about bone-binding antibiotic that we designed. As you know, there is high rate of morbidity related to orthopedic infection, and there are limited antibiotic options for prevention of this type of infection in the bone. And usually the systemic antibiotic for chronic cases fail to have response to this infection. And we were thinking about designing a new type of antibiotic, designing a new type of delivery for antimicrobial compound by using this concept that using one of the antimicrobial compounds such as cationic selective compound CSA90, which has a broad spectrum of antibacterial property against gram-positive and gram-negative bacteria and even their resistance form. And we wanted to actually bond this molecule, this antimicrobial compound, to another compound which has very high affinity toward hydroxyapatite. And our hypothesis was by doing this, it can directly go to the bone and it can show its efficacy against the bacterial infection. And bisphosphonate alendronate is one of the compounds which is used for treatment of metabolic bone condition, and it has very good affinity toward hydroxyapatite. Our idea was to bond these two compounds, and for this purpose, we used some chemistry background and use this linker to bond these two compounds together. The FTIR and NMR result demonstrate that it is possible to make this synthesis, and now we need to confirm that the activity of this antimicrobial compound doesn't change by this chemical bonding, and also the affinity to bond to the hydroxyapatite hasn't changed. We did a very simple study in here, in vitro study, that we added hydroxyapatite in a media and added this bone binding compound to the media as well. And we could see within 1 hour, the concentration of this bone binding antimicrobial compound was significantly dropped, which demonstrates that this new compound doesn't change this affinity to the hydroxyapatite. And the other formulation that we made with the CSA without having this bisphosphonate alendronate doesn't absorb to the hydroxyapatite. In here, we show that the efficacy or antimicrobial effect of the CSA also after bone binding after binding to the bisphosphonate alendronate hasn't changed significantly. Then we can use this material to directly deliver the antimicrobial compound to the bone. This product has been sub-licensed to N8 Medical Biosciences for commercialization, and hopefully it come to the market soon. The next product that we designed again for the bone treatment is engineering a sugar-based drug carrier with phase transition property. As you know, many peptides and protein already approved for the treatment of diseases such as cancer and also bone repair. A group of these peptides are, for example, BMP, which has a potent forming agent used to promote open fracture repair and many other applications. The current delivery of the BMP to the bone is by using collagen sponge, but because collagen sponge doesn't have good mechanical property and also has burst release, is not really very ideal for the delivery of peptides. And other system for delivery of peptides can result of degradation of these type of molecules. We recently used another compound, sugar acetate azobutyric, which we call them SAPE. This type of delivery carrier, when you inject it to the bone area or any other parts of the body, it can do the phase transition and release the compound. The problem with SAPE is hydrophobic and doesn't have very good interaction with the peptides, and we couldn't make a very good formulation from this product. We try to, our idea was to make another type of sugar, high viscosity sugar-based carrier, which can have better property and can actually have the same physical property, but have better interaction with the peptides. For this, we actually use a compound called deoxyribose. Is a precursor to DNA and a molecule which can stimulate endothelial cell migration. To make this deoxyribose, as high viscosity polymer that can be injected, we need to do the polymerization. And then to make it amphophilic compound that can have good interaction with the protein, we did another step to do the esterification to replace OH group with isobutyrate chain. And as you know, for sugar to make a polymer is very challenging because the sugar degrades very fast by temperature. To make the reaction easier, we designed a system that doesn't need to use that much organic solvent or toxic solvent. We just use glacial acetic acid, and we could do the reaction in this condition. And by controlling the temperature and the time, we could make different type of these polydeoxyribose, and the FTIR and NMR result confirm that this formulation can form. And we can make different viscosity of this solution, and also the burst, we don't see that much burst release in the in vitro model that we use. And basically, it has zero order kinetic release of the peptides. By confirming this, we make different formulation of SAPE with the BMP and also polydeoxyribose that we design with BMP2, and we could see that the bone can regenerate, and also suffering or staining show that cartilage can regenerate in this material, and also polydeoxyribose demonstrate to have endothelial causes endothelial cell to form, and observe, we could see that it has some angiogenic properties. And compared with the SAPE, it doesn't show that much immune response. In another study that we have conducted, we compared the bone formation close, when we inject this BMP with this carrier. And we could see that superior properties of polydeoxyribose compared with SAPE because of these amphiphilic properties that it has. It can interact with the peptide much better, and we could see that less variation of the bone volume formation in this sample of polydeoxyribose compared with SAPE, and we could see the bone can form in this material. Then polydeoxyribose is a very good candidate as a carrier for the peptide for injectable delivery. We work on designing injectable delivery for target delivery of drugs, and also for designing injectable hydrogel for different applications. In here, one of the examples is designing temperature responsive hydrogel, and we wanted to make some hydrogel which can be biocompatible, similar to the peptides and protein or other natural polymers, but have a good mechanical property similar to the synthetic polymer. And we wanted to have a material which can have tunable properties. To do that, if we wanted to make hydrogel and injectable, we have to use material which can be water soluble. And because we wanted to not use any other additive or agent, for example, to be UV cross-link or other type of chemical cross-linking, we want to design thermally responsive polymer. And we wanted to make sure that this polymer that we synthesize can bond to the peptides. For this purpose, we use Loctite to make it water soluble and bond to the other parts of the molecule that we plan to design. We add HEMA and NIPAM, which N-isopropyl acrylamide, and OECMA compound, and to make it bond to the peptides, we use NAS group, N-isopropyl compound. And this molecule can be formed, and NMR result confirmed the formation of this material. We could make 18 different formulations that were easily soluble in water, and by changing the temperature, we could make a gel formation. And we make different formulation of this material, and we found that it's also biocompatible because the cell could proliferate in this material. One of the formulation that we made is with thymosin, which is a synthetic peptide with no cell-specific phenotype response. And with this formulation, we call it TX140, and we wanted to optimize how much thymosin we need to have still good gelation property. And by this optimization, we consider to use 30 milligrams of this compound. And now, if we wanted to use it as an injectable formulation, we need to do several tests. And in here, we conducted several in vitro and ex vivo tests and in vivo tests to confirm that this material can be used as an injectable formulation. In this ship osteotomy model, we can see that we can inject this material, and even it is mixed and diluted with blood, it can still form the gel, and gel remain where we injected this material. In this another ex vivo bovine model, we could see that after we inject the material, even without any physical containment, under the dynamic condition, under 100 cycles, still the material remained where we injected the material. In this porcine heart tissue model, we could see that after injection of the material, even after 24 hours, when we keep the sample under dynamic condition, it doesn't still adhere to the surrounding tissue. And in this animal model, we could see that we can inject this hydrogel under the skin without using any physical containment, and even after 2 weeks, it remained where it has been injected. Then it demonstrates that this material can be used as an injectable formulation much easier and without using any sutures or any other physical containment. If you want to use it for clinical application, it's good to consider about safety of this material and make sure that we can sterilize this material to use it as an off-the-shelf product. For sterilization, we use gamma radiation, and we found that we can decrease the bacterial or sterilize it up to 10 power minus 6. And without having any issue. It doesn't have any effect on the properties of this polymer. And also we need to confirm that there is no organic residue solvent remain in this product. And by conducting several tests, we found that the maximum allowable limit, the concentration of the or residue of the organic solvent is significantly below the maximum allowable limit. And we found that it also doesn't have significant amount of endotoxin level. Then in terms of these properties, it is safe, but we also need to confirm that when we inject it in the body, the degradation or bioresorption of this material doesn't cause any effect on the or different organ in the body. For this confirmation, we have done a comprehensive study looking at subcutaneous injection of 2 milliliters of this sample in male and female mice model for 10 weeks. And we compare different doses of this material with a control sample, which was a saline solution. The data in here shows that the weight of the animal hasn't changed, and the weight of different organ hasn't changed after even 7 weeks, 10 weeks that we did this test. In here, we could see that we observed this material has some histolytic reaction compared with the saline solution. And in here, we see that after 4 weeks and 6 weeks, there is some angiogenesis property observed in this material, which these two confirm that this material has got no tissue formation and remodeling effect. And the data in here demonstrate that compared with saline solution, there is no significant effect on liver, kidney, and other tissues in our body in the mice model. This data confirm that this material is safe to be used for a human application. In here, before we do the clinical trial, we wanted to demonstrate that this material is suitable for soft tissue repair. And in this mice model, we injected this material in the left side, and in the right side, we injected the collagen as the comparison. And we could see, in here, we don't need to use any sutures, but with collagen, we need to have some sutures. And we compare these two materials for this wound healing under the skin for a month. And as you can see in this fluorescence, radiation, we could see that for the TX140, the material that we designed, compared with collagen, we can see more neovascularization formation in the TX140 compared with the collagen. And this, after 28 days, started to stabilize and to allow the maturation and subsequent progression of the wound healing. Then the performance of this material compared with collagen is much higher. This study demonstrates that this material is good for soft tissue repair. And this histology in here showed minimal inflammatory response for the TX140. For collagen, we see multiple layered inflammatory fibrous tissue formation, which within 4 weeks prevent the cell permeation to the tissue. And HS staining cross-section of this sample shows that we can see infiltration of host cell to the skin remodeling, and also we see blood vessel formation, dermal connective tissue formation, and also native collagen fiber formation in this material compared with collagen that was much higher. We conducted another animal study using sheep model, and we wanted to see whether it can regenerate the bone or hard tissue in here. The data in here shows that within, after 6 weeks, we use negative model, empty, positive model bone graft, and this was in TX was of a material, and we could see that within 6 weeks, the sample which has bone graft, of course, higher bone formation we observe in here compared with the other material, but after 12 weeks they were comparable. Basically we could see compact and trabecular woven bone formation in this TX140, and similar to what we see in the bone graft. And also the material, the region that is left the material was much smaller. Then this study also demonstrates that this material, this injectable hydrogel, can be used for regeneration of bone. Then we conducted some clinical study in here for socket preservation operation. And we can, in this operation, usually after the tooth is extracted, they add bioos, which is bovine samples. And after that, they have to put a film on the top and do the sutures and wait for 6 months to generate the bone to make the implantation. And sometimes this is also not successful, and they need to do reoperation and to fill the gap again with bios. We propose to use this injectable hydrogel after the tooth extraction. We can do simple injection, and this hydrogel can mix with the blood, and you can see that after the operation is much simpler, takes less time, and also within 8 days and also 28 days post operation, we can see the healing on the surface. And we conducted this test in 10 healthy participants, and in all these tests that we have done, we could see that there was no side effect observed, and we could see that the soft tissue is formed after 7 days of post-operation, and after 3 months we could see that the woven bone form in this material section that we injected this material. Then this demonstrates that this material can be used as an injectable hydrogel for bone regeneration. This material, PNPH or TX140, led to a startup company, Terasaki, which now use this material for different applications, drug delivery and wound healing and many other applications. Apart from designing hydrogel and material for tissue engineering and drug delivery, we are working on designing non-degradable material, which can be, soft, and elastic material with tunable mechanical property. The design of such material which has good toughness and elastic is very difficult and challenging, but we design an interpenetrating polymer network which can be easily synthesized by by mixing two different type of class of the polymers. The first class is HPU, hydrophilic polyurethane, and the other one is another polymer which can be cross-linked in the structure of the physically cross-linked HPU. And this polymer is produced by using the polyethylene, HEMA, and NAS. And the good thing about this design, we have lots of degree of freedom in here. We can change the chain of this molecule or this molecule, and by using different molecular weight, we can play around with the physical property and mechanical properties of this polymer. We call this polymer PFAN, and you can see in here that by changing the ratio of the cross-linker, we can have different mechanical strengths, and if you use a smaller amount of cross-linker, we can have lower swelling, stiffer material, and if we use higher cross-linker, we can decrease the Young modulus and toughness of this material. And this material is quite actually tough. You can stretch it as much as you want, and it can still come back to the original shape. And we can play around with the HPU length as well, and this can, by changing the length of this HPU, we can change the mechanical properties of this material. This material is also biocompatible and doesn't have any toxic effect. And the PFAN is also, we can, as I mentioned, we can change and tune the mechanical property. We can mimic the cardiac, muscle, tissue mechanical property, or spinal cord mechanical property. And also in here, we can see that we can have different toughness of this material. And basically, PFAN can be used for different applications, for soft robotic, for implants, and also for maybe designing sensors. I talk about sensors, a group of our researchers working on designing different type of sensors. And these sensors can be used for food application and also for checking the food safety and quality, or for diagnostic purposes. Mainly, we are working on designing gas sensors, which basically gas sensors can be used for testing the food safety and quality to understand whether the food is spoiled or not, which this type of sensor can give information in the supply chain for release of the product and prevent the food waste. And for this application, we were thinking about designing sensors which can be, colorimetrics. We can change the color, such as in here, these two examples of our study, or it can be chemoresistive or amperometric detectors or sensors. But our idea was to make sensors which can be small, miniaturized, and doesn't have rigid material, and it can be used, on the, for point of care application. In here, I just wanted to give you examples of one of our sensors that we designed for detection of peroxide, hydrogen peroxide. We make it very simple. I mean, we use a conductive material for detection of the hydrogen peroxide, and we add horseradish peroxidase to this polymer. But the problem is this material, P.PSS, which is broadly used as a conductive polymer, is very brittle, and we cannot use it for we cannot bend it or as a wearable sensor. We talk about PFAN and also hydrophilic polyurethane. We come up with this idea to blend this polymer with HPU, and hopefully we can improve the mechanical property and design elastic sensor. And this actually worked, and we could see that we can make blending of these two polymer, and the mechanical strength of this P.PSS after blend with HPU significantly improve. You can bend it as much as you want, and the resistance of this polymer, even we change the curvature, doesn't change, and it will make different cycle of stretching, and up to certain level doesn't change the resistance of this polymer, conductivity of this polymer. And we could design an amperometric sensor by using this material. And the presence of the horseradish peroxidase is important. If we don't add it, we don't see significant, good trend. And in certain range for the hydrogen peroxide, it has linear performance. And if we want to use it for this range, we can consider that it is a linear, and we can use it for measuring the hydrogen peroxide concentration. The good thing is the presence of other compounds such as glucose or ascorbic acid or fructose really doesn't change the reading of this hydrogen peroxide concentration. We test that at two different concentrations of hydrogen peroxide. First, adding this compound and then adding the hydrogen peroxide, we see that the reading is exactly similar to what we had before. And again, we increase the concentration of the hydrogen peroxide and then add this compound, the reading hasn't changed again. And it has a very good shelf life as well. And another thing is with this system, we can actually design a chemoresistive sensor as well, which has required less electrodes. In this case, we can use 2 electrodes, and we could see a linear range of detection for the hydrogen peroxide measurement. And this hydrogel can also have sensor can read very quickly within 10 seconds. Even it can work under high humidity without, humidity doesn't affect the reading of hydrogen peroxide. This was the problem that we faced when we use a P.PSS on a paper-based sensor. And we can use this sensor for broad range of application, chemoresistive, for different application in medical or for environmental. We can see that, we need different range of measurements. And for most range, chemoresistive sensor is working. And if we need higher range of hydrogen peroxide, we can always use amperometric sensor. Another example of our research is related to designing a patient-specific implant for children with congenital heart disease. Congenital heart disease is actually, is the most common congenital disorder in newborn infants, and nearly 1% of infants may born with this disease. And about 25% of these babies require to do operation for and in the first year of their life, which is very, very sad to hear that. And allograft or xenograft valve conduit are commonly used for treatment of this disease, and that they need to connect the right ventricle to the pulmonary artery pathway. And such conduit do not grow and have a size mismatch between the patient and prosthesis. And particularly when this initial implant is, used, they have to do several operations after that because they grow up and the size is not the right size. This allograft and xenograft may also cause immune response and fail. And the second issue is the pathway, as you can see in here, is not really a straight line. And most of the prostheses which are available is a straight line. And therefore, during the operation, the surgeon required to do an additional operative customization for each individual anatomy of the patients or these children. And this results in making a non-ideal pathway for the blood flow. And basically, we can have more energy loss and biomechanical stress on this valvular conduit, which results in dysfunctioning of this conduit and then failure of this conduit and another operation or even led to mortality. Our research team is very interested to improving solutions that hopefully improve these prostheses that are used for implanting this valvular conduit. We think about using food industrial designing personalized heart valve by basically using imaging and then geometry construction, computational modeling, material development, and additive manufacturing to hopefully making the better implant for this purpose. And for our study, as I said, we did MRI for several patients. And in here, I show you about 6 of these patients. And you can see that each of these patients, these infants, have different structure for the pulmonary, this conduit that we need to design for them. And we can see that how important it is to make personalized tissue. For personalized implants, sorry. And we use the MRI result of each patient and constructing a 3D model by segmentation on this MRI, and then construction of the geometry by interpolating between these extracted cross sections. And then we make geometry ready for simulation, and we use that for our simulation and see what will be the best shape of the conduit that we need to make for this specific patient to minimize the energy loss and wall shear stress on the conduit. And to use the computational fluid dynamic, we also use velocity from an echocardiogram of each specific patient. And by collecting this information, we try to make different models and use different configuration and to see whether we can decrease the shear stress and basically, in here, we can see the total pressure loss can be decreased. Okay, in here, we can see average wall shear stress. We can design different models to decrease that. And by doing this, we can see that we can decrease the wall shear stress on different for each patient. We design different model and we can decrease the wall shear stress significantly. And also energy loss, for example, for patient A, by designing better modified conduits, we could have less energy loss. And we hope that by doing this, in here we can see that how much the stress on the wall change and how much the deformation if we use the original shape or and also the modified form. Now, this conduit need to have a valve also. We first use this conduit without the valve, but now we need to add the valve as well. For the valve, they are, and also conduit, they have different approach. Some people use tissue engineering approach. And there is one sample of, a study that company Zeltis produced a electrospin product. And but still, they have seen a calcification of the leaflet, and I haven't seen a successful, clinical trial from this sample. And some researchers working on better design of the valve using 3D printing, and they have achieved, but they haven't still put it in the clinical study yet. The most successful so far, I have seen a study that they use a non-tissue engineering approach. Basically, they design heart valve from Foldax company. They call it Tria valve. And this valve is made from siloxane polyurethane uroelastomer. And they have confirmed that with this material, which is quite inert, they don't see panels formation, while the current material which is used causes this problem. We are thinking about biomimetic design of the valve and not using the flat leaflet. To do that, the same approach that we take for the designing the conduit, we are working on heart valve. We take the heart valve of the animal at this stage, do the macro CT scan, and make a similar stage that we designing a 3D CAD model, and put that in the computational modeling using the mimicking the modulus of the native heart tissue. And we design different structure of this heart valve and use the computational modeling. And now with this model, we can see whether each design of the valve can open or not. And we can look at the deformation of the valve and also stress that is on the valve. And basically by this modeling, we are able to test and design optimum shape to minimize the deformation and minimize the stress on the valve. And by doing that, we hope that we can increase the durability and stability of this heart valve and minimize the number of operations that these children need to go through. So far, we have been able to design valvular conduit using biocompatible siloxane polyurethane, and with the optimization that we have done with the computational modeling. And with the pulse duplicator, we could confirm that this valve can operate. And we compare the performance of this valve with the standard valve, which is used for the pulse duplicator. And while this diameter of a valve was larger, however, the fluid flow percentage which was passed through was still below 20% and very comparable with this material or the valve that is already used in this device. And this gives us hope that if we can improve the design and make the personalized design, hopefully we can decrease the regurgitation and stenosis, which are the common problem for this heart valve and valvular conduit system. And we hope that we can give hope to the children who has this type of disease in near future. Now, shift the research that we have done from biomaterial to area of food. We are working on designing plant-based food products, and the driver behind the plant-based protein or food product is people are looking at having healthier habits. They wanted to consider about animal welfare and also sustainability. And we know that our population is growing, and within by 2050, we will have 11 billion population, and animal farming will not be sufficient because it will use 80% of the land and producing only 16% of the food or meat or protein that we need. And we will pass the planetary boundary, and we need to consider about designing new sources of the protein for population. We work with the algae food company that supported our research to design plant-based food products, and by conducting this research, we could use that. We can use protein from plants, and I mean there are so many companies so far working in this area, but the plant-based which is produced from this research demonstrate that it has very superior properties in terms of texture and flavor. When we did the sensorial testing, the performance was better than current product in the market. This material is already commercialized, and hopefully it can be imported to US or you come to Australia and test it. And there are other approaches for production of the protein that we can use our tissue engineering expertise. And we all know that this is very expensive, but hopefully in near future, the cost can be decreased for production of the protein and fat and other tissue using the cellular agriculture. And even the cost is very high, but some of the company claim that they can produce this type of product by cellular agriculture, and many researchers now working in this area. What we are doing in our center, looking at cellular agriculture, but we're using insect cell. Why we are going to use insect cell rather than stem cell? Because they don't require CO2. We don't need to control the temperature to 37 degrees. And they are resilient to shear, pH change, and osmolarity. And also, we can use other sources of the nutrient, and cheaper source of nutrient for growing this insect cell. And the insect cell, actually, they can insect protein is quite good quality in here, and the amount of fat that they can produce is much lower, and we can produce high quality protein using different type of insect cells. Our preliminary results show that within 4 days we can produce a significant amount of the triglyceride from this, and if we use this technique, we can actually make omega-3 and high quality protein which are soluble and as well. And this is the beginning of our research, but we are working, and hopefully in near future we can have high quality protein and fat produced by this cellular agriculture system. I would like to thank the organizer of this workshop and also seminar series, and also our collaborator, and also organizer that support our research, and industry partners that collaborate with us. And thank you for your attention. And if you have any question, I would be happy to answer.
Thank you. Thank you for the great talk, Fariba. I truly appreciate it. Covered a big, massive area of research. One question is, have you improved the functional and structural properties of applied biomaterials in your studies? Or did you just use them in native form?
Sorry, which material?
This was at 2:30. So I think you mentioned that K140, and you mentioned
yes, the antimicrobial compound. Yeah. Yes, that antimicrobial compound, we just conjugated to or bonded to the bisphosphonate alendronate. Yes.
Okay.
The question was,
did you use the materials in native form, or did you improve their functional structural properties?
No, we use material as it was and then chemically bonded to the bisphosphonate alendronate. Yes.
Thank you. In your bioabsorbable material that degrades to carbon dioxide, why is starch also blended? How does the mechanics change during degradation? Could it lead to implant failure if it's load-bearing application and bond?
Yes, the reason, I mean, this polypropylene carbonate, to make it, to improve the mechanical property, we blend it with starch. And also when we blend it with the starch, it can have better integration. We compare pure PPC with PPC starch. We could see that it better integrates with the surrounding tissue because of its water absorption and swelling properties it has when it absorbs the water.
Thank you.
And also we could control the degradation rate, yes.
All right, thank you. For your regeneration biomaterial compared to collagen as well as bio-osseointegration, why would you expect your material to have a duality to form bone and soft tissue?
We wanted to demonstrate that this material as an injectable can be used for different purposes, not just for soft tissue. We could use it for hard tissue as well, for broad application. Actually, we use it for even, as I mentioned, nasal drug delivery and, many applications.
I see, thank you. One other question is, you mentioned biosensors. You're also implanting materials for the wound and other areas. Do you make sensors that can detect whether there is biocompatibility or contamination or infection in your, do you combine, biosensor research and your regenerative medicine research?
This is a very good question. Actually, we have received recently a grant. And this is what we are, definitely we consider about biocompatibility, but for, food application, we need to be very, very careful that material that we use is not toxic for that purpose. If we wanted to use it externally for diagnostic purpose, of course, biocompatibility is not an issue, but for wearable and other application, we definitely consider about biocompatibility. And so far, material that we use are biocompatible and doesn't cause any issue.
Thank you. Thank you. One final question is, you mentioned about insects because, the cellular agriculture is a big deal. In the world of insects, you showed many insects. Which one is the low-hanging fruit to make the, meat or meat-inspired things?
Yes, I think we need to really work in this area. I cannot say which one is good, but we need, we started this research about a year ago using insect cell agriculture for cellular agriculture. At the moment, we use standard, but we are thinking that this can be, very important to use different type of cell and screen them first and see which one faster growth. If we wanted to commercialize it, we have to consider about optimization. And we are working in this area actually. And several other groups in US also working in this area.
How about fish?
Fish also possible, yes, but yes, some, as I mentioned, in not just fish, some other mollusks and has been used in New Zealand and also in Singapore. They do research in this. For example, they use prawn tissue. But as I said, the issue of using fish or prawn, again, those tissues sometimes you need to control the temperature. Compared with insect cell, is more challenging.
Well, thank you so much for the fantastic talk. Truly appreciate it. Lots of interesting questions. Thank you for your time.
Thank you.
Have a nice day.
Bye.